We're excited to have Dr. Juan Latorre give today's lecture. Dr. Latorre received his medical degree from the National University of the Northeast in Argentina and received a PhD in pharmacology from Tulane University. Dr. Latorre is currently an adjunct professor of medicine at Duke University and Louisiana State University. Previously, Dr. Latorre was a faculty member at the Clinical Pharmacology Center, Northwestern University. Between 1981 and 2006, he was professor of medicine and pharmacology at Tulane University School of Medicine. Dr. Latorre joined the NIH Clinical Center in 2006 as the director of Clinical Pharmacology program and led this course, The Principles of Clinical Pharmacology, until his retirement from the NIH in 2016. Please enjoy the presentation.
Welcome to Principles of Clinical Pharmacology. My name is Juan Latorre, and today I will present an overview of the discipline of clinical pharmacology, and I also will introduce the basic concepts in pharmacokinetics and its clinical applications. The focus of the course traditionally has been on the scientific basis of drug use, development, and evaluation. We do not consider this to be a course in therapeutics, but of course there will be relevant examples of applications of clinical pharmacology in therapeutics. We will discuss general principles that are applicable to both old and new drugs. There is a textbook that has been used for this course for a number of years, Principles of Clinical Pharmacology. The lead editor is Dr. Arthur J. Atkinson, Jr. So, let us see an outline of what I would like to cover for you today. In the first part, we will have an overview addressing the general scope of the discipline, some brief historical notes. We will talk about what do clinical pharmacologists engage in as professionals. We will emphasize the topic of variability in drug response as an area of great interest in our field. Also, adverse drug reactions and their impact both in terms of drug development and clinical use of drugs. And finally, a brief overview of drug development. So let's move on then and define pharmacology as the study of drugs and biologics and their actions in living organisms. Generally, when we talk about drugs, we think of small molecules, chemical agents. When we talk about biologics, we're thinking about large molecules, peptides, and antibodies. The most basic definition of our field is that clinical pharmacology is the study of drugs and biologics in humans. The discipline really spans the spectrum of drug discovery, drug development, drug utilization, and drug regulation. We aim, in clinical pharmacology, we aim at advancing therapeutics in humans with mechanistic understanding of drug actions. This is an area termed pharmacodynamics, and also drug disposition, and that is, of course, the subject of pharmacokinetics. Now, you know, of course, the concept of translational sciences and how much it has been emphasized for the last decade or so. Basically, we talk about knowledge that has been acquired in animal or in silico models of disease or through ex vivo studies in human tissues, or in vivo studies in healthy or diseased humans, that then is translated into effective treatment for patients. Clinical pharmacology is a translational discipline essential for drug development and therapeutics in humans. Now a bit of history focusing on the founders of American clinical pharmacology. I'm talking about Drs. Harry Gold and Dr. Walter MODELL at Cornell University. And this is a partial list of their accomplishments and fundamental contributions, introducing the double-blind clinical trial design in 1937, initiating the Cornell Conference on Therapy a couple of years later, and in the early 50s, analyzing digoxin effect kinetics to estimate the absolute bioavailability as well as the time course of the chronotropic effects of digoxin. We'll come back to this example later in the talk. And in 1960, they founded the journal Clinical Pharmacology and Therapeutics, which is today, of course, a leading journal in the discipline. Now, at the NIH, we should mention Dr. Albert Schertzma, who headed the experimental therapeutic branch at the National Heart Institute from 1958 through 1971. He trained individuals of the stature of Luke Gillespie, John Oates, Leon Goldberg, Richard Kraut, Ken Melmon, and many others that subsequently became leaders in the discipline as well. Their research focused on serotonin and the carcinoid syndrome, pheochromocytoma, antihypertensive drugs, and many other contributions. Now, what are the professional goals of clinical pharmacologists? Well, we are interested in the discovery, development, evaluation of new medicines and how their use is regulated by the Food and Drug Administration in the United States and other regulatory agents in other countries. We are also interested in optimizing the use of existing medicines and often finding new indications for all drugs. But as I mentioned in our initial outline, a critical area of interest to clinical pharmacologists is to define the basis for variability in therapeutic and toxic responses to medicine. And this is an example looking at the exposure to two anti-diabetic drugs, pioglitazone on the left side of the slide and metformin on the right-hand side. And we're looking at drug exposure in terms of the area under the plasma concentration time versus time curve. And this is an AUC, area under the curve, that has been normalized to a 15 milligram dose of pioglitazone and a 500 milligram dose of metformin, and also normalized to 70 kilograms of body weight for a human patient. And you see the great variability that we see in drug exposure both in females and males in the case of both of these anti-diabetic agents. So that's one of the challenges clinical pharmacologists face in trying to understand the basis for this variability in drug exposure and how it may impact on the therapeutic actions of the drug. Another source of variability in drug exposure may relate to underlying genetic variants. In this instance, we are using the example of Nortriptyline, a tricyclic antidepressant that has been in use for many years, and the impact of cytochrome P452D6 polymorphism. And here we are plotting plasma concentration of Nortriptyline after a 25 milligram dose over time. And then we see the impact of the number of functional genes for CYP2D6. The first curve on top indicates a higher exposure for an individual that does not express CYP2D6 and actually by definition is a very slow metabolizer of this drug, and then the progressively smaller area under the curve with increasing numbers of CYP2D6 functional genes. This over here at the bottom is an individual with 13 copies of the gene that is also an ultra rapid metabolizer of this drug. So another source of variation in drug exposure and of course potentially on therapeutic efficacy of drugs in terms of this pharmacogenetically determined variation in drug exposure. Now let's turn to another major area of interest in clinical pharmacology, namely adverse drug reactions. Some toxicities of drugs can be managed and may be acceptable based on a risk-benefit ratio, but other adverse reactions and toxicities by their nature and severity are really unacceptable, and those drugs either have to be removed from clinical use or used with great caution and adherence to significant and close monitoring of the patients. We need to understand, of course, that risk-benefit is contextual, depending on the drug and the disease that we intend to treat. It is not the same to consider potentially serious toxicity for a drug intended to treat hypertension, which is a condition that needs lifelong therapy, compared to, say, treatment of cancer, a disease that is potentially lethal over the short term and that requires very intense treatment with combination of drugs that have very significant toxicity. So again, risk-benefit is contextual, and we must consider the drug in question and the disease that we are intending to treat. Now again, in terms of genetics, as it may relate to severe drug toxicity. Now this is a condition or situations, if you will, where an underlying genetic variant may predispose individuals to severe toxicity from drugs. Here we have the examples of HLA-B5701. Individuals that carry this HLA variant are at very high risk of abacavir hypersensitivity. Abacavir is a drug used in the treatment of HIV infection and AIDS, and prior to instituting treatment with abacavir, every patient is first tested for this variant HLA-B5701. If they have the variant, they cannot be treated with that drug and an alternative must be found. The next example that we show here is that of HLA-B1502, predisposing to severe carbamazepine-induced Stevens-Johnson syndrome. This is a serious cutaneous adverse drug reaction that actually can be fatal. So once again, the underlying genetic variants conferring predisposition to severe drug toxicity. Another example of unacceptable drug toxicity is that of torsade de pointes. What we're showing here is an electrocardiographic record of heart rhythm in a patient that suffered from an episode of this polymorphic ventricular tachycardia. This is a very abnormal rhythm. You can see here a normal beat, if you will, in the electrocardiogram preceding these runs of polymorphic ventricular tachycardia that is actually drug-induced. So this is another example of a potentially life-threatening adverse reaction from drugs. And here I'm showing TERFENADINE, which historically was the first non-sedating antihistamine that was introduced in the United States market under the brand name of Seldane, but was subsequently withdrawn from the market because of the risk of drug-induced arrhythmias. Now look at the metabolic transformation of TERFENADINE in humans and the production of terfenadine carboxylate as a metabolite. Very interestingly, this metabolite is active. It also has this antihistamine pharmacological action, and it's also a non-sedating antihistamine, but terfenadine, which is marketed as Allegra, does not have the risk of a drug-induced arrhythmia like torsade de pointes. And this again brings us to consider and remember the importance of studying drug metabolism and assessing whether metabolites are also pharmacologically active or are otherwise inactive once biotransformation has taken place. Let me bring you the example of thalidomide. Again, in terms of unacceptable drug toxicities, but actually with a very interesting history, as I will show you in a moment. Thalidomide was introduced in the 1960s as a sedative and actually was prescribed as an anti-nausea medication to pregnant women. Unfortunately, in many countries, although not in the US, because thalidomide was not approved in the United States and actually was not allowed to enter the market at the time because of the discovery of some severe toxicity to unborn children due to prenatal drug exposure. This led to an epidemic worldwide of PHOCOMELIA, children born with severe defects in terms of their limbs, and of course this is a very unfortunate outcome of the use of that drug in pregnant women. Now there were consequences to this thalidomide crisis. For one thing, the United States Congress approved the Kefauver-Harris amendments in 1962 that instituted new and more strict FDA regulations to establish whether drugs were, on the one hand, effective but safe, and the process that has been modernized over the years, but again emphasizing safety and demonstrating efficacy of drugs before they're allowed into the market. The Institute of Medicine and the National Academy of Sciences began to review therapeutic claims at that time, and also more research on the causes of adverse drug reactions was encouraged. And the National Institute of General Medical Sciences created a number of clinical pharmacology centers in the United States to again, implement rational drug development to establish the scientific basis of drug use in clinical medicine. And again, sadly, as a consequence of this major thalidomide crisis. So our discipline is eminently involved in the development and evaluation of new drugs. We start with drug discovery, and this is a process in itself that we will be addressed in detail in another session of this course. Then we have preclinical, meaning animal testing of candidate drugs, and eventually clinical evaluation to demonstrate safety in humans and whether or not the drug is effective in a given clinical condition. But then we also have post-marketing studies. Once the drug enters the market, we continue to evaluate for the possibility of rare adverse drug reactions that were not discovered in the pre-approval stage, and also performing studies in special populations like the elderly and in children. Now, this is a schematic of pre-marketing drug development. You see here the phase of preclinical development. We have animal models, we have assay development, we study pharmacokinetics and pharmacodynamics in animals. We, of course, begin to study animal toxicology in the short term and the long term if the drug is intended for chronic use. And once a package of information is developed that indicates that the candidate drug may in fact be promising, an investigational new drug application, the IND, is filed with the Food and Drug Administration or other regulatory agencies, and then we begin the process of evaluating drugs in humans, typically considered as phase 1, first dose in human studies, dose escalations to assess tolerance, phase 2, when we do the proof of concept studies, treating patients with the condition that may benefit potentially from the drugs, and phase 3, the large randomized clinical trials comparing the new drug to a placebo or to a previously established therapy. And that then leads to the submission of a new drug application, or NDA, where the sponsor asks the regulatory agents to review this body of evidence and request approval for marketing the drug and to begin using the drug in clinical practice. One way to look at the phases of drug development is with the learn and confirm paradigm. The late Dr. Lou Shiner and his colleagues advocated this approach. Phase 1 and phase 2 are the learning phases of drug development. Phase 3 is the confirmatory phase. And phase 4, again, is the post-marketing phase, but learning continues, focusing on rare adverse drug reactions and special populations if required. Now, let's talk for a moment about drug repurposing. This is an area where the National Institutes of Health and other academic investigators have been very interested in, and that has to do with finding new biological targets and new therapeutic indications for all drugs. What are the potential advantages of this approach? Well, for one thing, it may shorten drug development time. We already know a lot about the safety of the drug, and we also have data in terms of the human pharmacokinetic behavior of the drug. And drug repurposing then, and this is the concept of Dr. Austin at NCATS, is illustrated in this fashion. Now, typically we have a process of drug screening of thousands of compounds, and the whole process may take 10 years between identifying the target agent and performing all the preclinical and clinical phases of drug development that may then lead to drug approval. What if then, through repurposing of a much smaller number of drugs that have been in use for other indications, could perhaps shorten the period of drug development to a couple of years. Now, this is ideal, but conceptually, again, very important. And we do have examples of a number of drugs that have been repurposed. And very interestingly, we have again thalidomide, extremely toxic and forbidden in pregnant females. But nevertheless, through the clinical observation of a physician in the 1960s, it became a very useful agent to lead, or rather to treat, a complication of leprosy called erythema nodosum leprosum. Again, a drug that otherwise was banned from marketing becomes now useful in a clinical condition like erythema nodosum leprosum. Years later, the drug was actually studied in the condition of multiple myeloma, again a form of cancer, this time through targeted drug development. In any case, these are now two FDA-approved indications. This is an immunomodulatory agent. Marketing is done under a very special and very restricted distribution program referred to as System for Thalidomide Education and Prescribing, but a very good example of drug repurposing. And in this slide, I show you a list of drugs that were approved originally for a different indication, but now are FDA approved for indications that, for example, for sildenafil, include pulmonary hypertension, lamotrigine being used for bipolar disorder, and so forth. So again, repurposing as a viable and potentially very important way to look at finding new indications for all drugs.